Hello and welcome to our webinar, The Skin Microbiome and Labskin, the next frontier in skin science. I'm Dr. Nicola Kingswell, Scientific Director at Labskin Limited, and today we're going to explore the composition of the skin microbiome, why it's important to us, and some study tools. As we all know, skin health is multifactorial. It's not just the products that we put on our skin that can affect its appearance. Biological factors, such as the skin microbiome, interplay with the other organs of the body, such as the gut and the brain, your genetics and ethnicity, hormones, especially for women with puberty, menstruation, pregnancy and the menopause, and the immune system can all affect the skin. Environmental factors, such as exposure to the sun and UV radiation, pollution if you live in urban environments, and humidity, dryness, air conditioning or cold exposure all play a role in skin health. Lifestyle factors such as the food you eat, the supplements you take, the hydration and water intake, the exercise you do, the stress you are under, the quality of the sleep you get, and the products that you use on your skin will all affect its healthy appearance. The skin microbiome has an important role to play in skin health. It can regulate the pH of the skin, prevent infection from pathogenic bacteria, viruses and fungi. It can modulate the immune system and help fight off harmful bacteria. However, when the skin microbiome is out of balance, it shows in the skin by increased acne outbreaks, rosacea or eczema, increased dryness or flakiness, more skin infections or premature aging and fine lines. The skin microbiome consists of a diverse community of bacteria, fungi and viruses and other microorganisms that live on the surface of the skin. They work together to provide a vital ecosystem to maintain skin health, to improve barrier against pathogens by regulating the immune responses and helping contribute to overall skin hydration. The major player in the skin microbiome is bacteria, and we'll dig into some of these bacteria later on in a bit more detail. There are approximately 1 trillion bacteria living on our skin. That is more than the 8 billion people living on Earth, but less than the 100 trillion bacteria living in our gut. And there are both good bacteria and bad bacteria present on our skin, so commensal or pathogenic bacteria. Yeast and fungi such as Malassezia, the key microbe responsible for dandruff, are other community members. They help remove dead cells and other organic matter such as oils from the skin. They are mainly found in moist areas of the body such as between the toes, the groin, the scalp, behind the ears, the crook of the elbow and between the eyebrows. The most common types of virus found on human skin are bacteriophages that infect skin bacteria. These viruses have an important role in regulating the skin microbiome. In fact, bacteriophages are being studied in other aspects of human health as a way of targeting bacterial infections. Other viruses found on the skin include herpes, which can cause warts and verrucas. Other microorganisms such as mites can also be found on the skin. Demodex mites can be found in hair follicles where they feed on dead cells and oils. But if they grow in large numbers, they can exacerbate pre-existing skin conditions or cause a skin condition known as dermatodichosis. A balanced and healthy skin microbiome is important to overall skin health and disease prevention. 70% of skin conditions can be linked to an imbalanced microbiome. So these are skin conditions such as acne and eczema. People are 4 times more likely to develop eczema if their skin microbiome is unbalanced. Wound healing is improved if the microbiome is balanced. It is less likely to experience wound infections and slower healing rates. And approximately $1.5 billion each year is associated with treating skin diseases such as eczema and acne associated with microbiome dysbiosis. So what are the effects of an unhealthy microbiome? The initial effects can include spots or infections with redness, swelling and discomfort. Short-term effects can trigger eczema, psoriasis or sensitization, or even acne flare-ups. And the long-term effects include persistent skin diseases and has also been associated with prolonged susceptibility to skin infections and even a role in skin cancer. There are a number of tools that can be deployed to study the skin microbiome. So these include DNA sequencing, such as qPCR or RNA sequencing, which allows scientists to identify and quantify the microbes present to reveal the composition and diversity of the microbiome. There are culture techniques, so using shake flasks or agar plates with selective media to study the microbiome growth patterns, interactions and characteristics to provide insights into their role in skin health. And also bioinformatics, so new data analytics and even AI to analyse complex data to better understand microbial functions, interactions and impacts. Both good and bad bacterial species, so commensal or pathogenic bacteria, play a role in the skin microbiome. Most of these bacteria are common to everyone's microbiome, but their compositions can vary. So Cutibacterium acnes. This is found on oily surfaces and hair follicles. They feed on sebum produced by the sebaceous glands, and they can cause acne breakouts when they multiply in the presence of excess oils and block pores. So Corynebacterium, both commensal and pathogenic. Some can produce a toxin that leads to diphtheria. However, on the skin they are mostly harmless and are essential to maintain a balanced microbiome. And Staphylococcus. So Staph epidermidis is one of the good actors in the skin microbiome. It produces antimicrobial peptides that keep other microbes under control. It has also been shown to modulate the inflammatory response in the skin. It can ferment glycerol in products to produce succinic acid, which in turn inhibits the growth of Cutibacterium acnes. By contrast, Staphylococcus aureus is often considered a bad actor. It is found in the nasal cavity and respiratory tract, and it is spread via physical contact. This bacteria can cause staphylococcal scalded skin syndrome, cellulitis or eczema, and is often found in biofilms in non-healing wounds. Other bacteria include Micrococcus luteus, which is usually considered a good guy and beneficial to the microbiome, but can become pathogenic under certain environmental conditions. Lactobacilli are a well-known microbe species with beneficial effects in the gut and vagina. They have also been shown to have beneficial effects on the skin, and the absence of this bacteria on the skin has been linked to atopic dermatitis and psoriasis. So digging into the skin-gut axis a little bit more. We know that the gut microbiome has been studied for many years, and the influence of gut health on skin health is linked. And dysbiosis of the gut microbiome can trigger systemic inflammation, which can present as acne, psoriasis or eczema. Leaky gut syndrome allows toxins such as LPS to enter the bloodstream, which can disrupt the skin barrier, which presents as increased skin infections or premature signs of aging. And clinical trials using probiotics such as lactobacillus and bifidobacterium have shown positive effects on the skin, reducing acne severity and skin inflammation. The overuse of systemic antibiotics, high sugar diets and high stress lifestyles have all been shown to wipe out beneficial gut microbes and present in the skin as acne flare-ups, dullness or hypersensitivity. We are all aware of pre- and probiotics such as cultured supplements or fermented foods such as kimchi and sauerkraut to maintain a healthy gut microbe. It is likely that pro- and prebiotic ingredients in skincare will become more commonplace. And linked to the skin gut axis is the skin-brain axis, and this is a circular feedback loop. The brain signals to the skin via the endocrine, nervous and immune systems, usually as stress responses. Equally, the skin signals to the brain via the same systems in response to pain, irritation and inflammation triggers. The psychology of skin health is also important. When you look good, you feel good, and vice versa. which is why wellness rituals around taking care of your skin are so important to mental health. Psychodermatology will become more readily used in skincare, with the interplay between physiological and psychological featuring more in skin health. So what is Labskin? Labskin was developed at the University of Leeds back in about 2009 by skin microbiologists. They were interested in looking in the skin microbiome, but they wanted a better medium to understand how the microbiome interacted with the skin. So they developed the Labskin product. It consists of human primary fibroblast cells and human primary keratinocyte cells. And we have a specific method for developing the structure of the skin. It takes about 28 days to grow, just like human skin. And we, as a company, work with industry-leading skincare and cosmetic brands to support new product development, new active ingredients, to support screening and claim support. One of the unique features of Labskin is the ability to fully support, not just put on the surface, but to culture the skin microbiome. When we look at how Labskin compares to human skin, so we have human skin in the left panels or in the top row, we can see that the Labskin produces the same protein markers as human skin. So proteins such as cytokeratin, filaggrin, which is important in the skin barrier, laminin, procollagen, which is important as a precursor to collagens in the skin. But importantly, we have antimicrobial peptides such as beta-defensin and RNA7 that are expressed. This provides an excellent tool for studying the skin microbiome. So we can take blends of microbes that are found in the human microbiome and place them onto Labskin to look at how products interact with the microbiome, but also how that changes in the microbiome interact with the skin. So we can look at markers for inflammation, irritation. But we can also take swabs from human volunteers and put whole microbiome samples onto the skin. So these are some images of microbes on the Labskin surface. So in panel A, we have what we call our 3X mix. So this consists of Cutibacterium, Corynebacterium and Staphylococcus. These are the three main microbes that are found in everyone's skin microbiome. And you can see on the surface of the stratum corneum there that we do in fact have the microbes present. In B, we have Staphylococcus. So this is an S aureus that's on the surface of the Labskin. And in C, we have Malassezia globosa. So this is the yeast, the fungi that causes dandruff. And again, we are able to culture this on the surface of Labskin. Because of our experience in culturing skin microbiome the skin microbiome on Labskin, we have developed this microbiome-friendly certificate. We apply the microbiome microbes onto Labskin. We allow them to establish that consortia, and then we apply products directly to the surface. So these can be new active ingredients, these can be final formulations or anything in between. And then we look at how these products affect that changing composition of the microbiome. So we'll look at how it interplays with each of those components, but we also look at how it affects the skin. So we look at markers for inflammation. So if we get that dysbiosis of the microbiome, we may see irritation and inflammation in the skin. And we have validated acceptance criteria around these features. And providing that products meet our strict criteria, we will apply this microbiome-friendly seal of approval. So this is a case study where we took a whole microbiome swab from a skinfluencer. So last year I was approached by the BBC to feature on Morning Live. They wanted to do a feature on adult acne, so they had a couple of volunteers who were interested to see how their acne was caused by changes in their microbiome. So I jumped on a train with my lead scientist. We hightailed it from York down to London, where we swabbed these skinfluencers by the flower stall in King's Cross Station, and then we took these whole microbiome samples back to the lab. So this is just a look into the workflow that we did. So we took our Labskin samples, we got the microbiome sample, and we placed that microbiome onto Labskin. We allowed it to incubate for 48 hours, and then we applied either a negative control of DPBS only, a positive control, which in this case was acnicide, which is 5% benzoic acid, and we applied a test item. In this case, the test item was the product that the skinfluencer was using to control her acne outbreaks, and it contained 2% succinic acid. We placed that product onto the surface for 48 hours, and then we did enumeration of the microbes that were present. So we looked at how those products changed that whole microbiome composition. We did some histology, looking at any changes in the skin morphology, and we looked at IL-1 alpha, a marker for pro-inflammatory marker for irritation and inflammation in the skin. Alongside this, we ran a uncolonised Labskin sample, so this didn't have any of the microbiome on there, so that acts as our negative control. The results were very interesting. So for the microbiology, we were only looking for the Cutibacteria acne species present in the whole microbiome sample. So you can see in the initial inoculum there, in the initial microbiome sample, there was just over 10 to the 4 colony forming units present. With the negative control with the DPBS, we did see that increase in growth, which is great because that means that they are thriving on the Labskin surface. And obviously DPBS has no components there that will control that growth. With the positive control, the acnicide, the benzoic acid, we did get that reduction that you would expect to see. But with the test item, with the succinic acid, we got a much bigger drop with the compared to the positive control, the gold standard. So this was really interesting. So it was great for the skinfluencer as well. She was like, well, I've obviously picked the right product to control my acne on my skin. However, when we looked at the inflammatory markers, you can see here that the test item produced much more IL-1 alpha in the skin compared to the positive control. We wouldn't expect to see any with the uncolonised control, so there's no microbes on the surface. It just shows that the Labskin itself does not produce any IL-1 alpha or minimum amounts of IL-1 alpha. The negative control, we would expect to see that increase because we got that increase in the Cutibacterium species in the whole microbiome sample. So we've got that dysbiosis, which in itself will trigger inflammation. But the fact that the test item produced more IL-1 alpha just goes to show that you can't just look at a product's effect on the microbiome. You have to look at it in the host reaction as well, so how that microbiome interacts not just with the product but with the skin as well. We have a very large database of whole microbiome profiles. So we have collected facial swabs from a broad range of people that we then subjected to RNA 16S sequencing, so we could look at the microbiome present on their skin. And this is where when I said earlier about the three key players that are present in everybody's skin, this is where we get that information from. However, from that database we can see there are up to 500 microbes present in some people's whole microbiome samples. We're just starting to unpick some of that data, so this is just a little bit of a taster into what we have found. So if we look at a healthy microbiome, so these are people that have a balanced microbiome profile, and we can look at male versus female volunteers, and you can see straight away that there are some differences, and these are just the top 20 microbes that we're looking at. So these are the top 20 most prevalent microbes. So there are subtle differences between male and female. And then if we look at people who have an acne phenotype, you can see there are massive differences here between a normal balanced microbiome and an acne microbiome, and some of this is very statistically significant data. So we were starting to delve a bit deeper and pick into this a little bit more, because there's plenty we want to look at with this data. So what next for the skin microbiome? There's a lot more research that needs to be done into its composition and relevance. So compared to the gut microbiome, we're just sort of on the start of our journey into the skin microbiome and how we can influence it. So I think we'll start to see a lot more new products aligning with microbiome balance. So looking at things that care for and nurture that skin microbiome, but also products that can influence how the microbiome composition becomes balanced. So things that will purely target acne bacteria, or will work with other bacteria on your skin to change that microbiome, to control overgrowth of certain bacteria. And there is a possibility as well that you may see more skincare personalised to your own microbiome. And I've talked to a few people that are interested in this, that they will take a swab or sequence the microbiome present on your skin and then tailor a skincare regime specifically to the needs of your own skin microbiome. And what next for Labskin? So the microbiome will always be at the heart of everything we do. It's the basis for our technology, so being able to understand the skin microbiome was how the company was founded, and that will always be what we're interested in. So we want to do more research into the skin microbiome and what we can do to push our understanding and work with clients on their products to help support that skin microbiome. So the skin microbiome and Labskin will always be at the heart of this company. But also we're looking at new innovations as well that are not just around the microbiome, although there's a lot we want to do around the microbiome. So looking at things for different exposures to, like say, cold or dryness, looking at creating different microbiome profiles. So we have a healthy, balanced microbiome, but can we create more dysbiosis types? Can we mimic atopic dermatitis or psoriasis with a microbiome? So we have new models coming through for atopic dermatitis as well. We have our pigmented model, so Labskin M with melanocytes. And we're just starting to dig a little bit into research, and there are a couple of interesting journal papers out there that show that different ethnicities with different melanocytes have different microbiome profiles. So can we mimic that in our lab? And if so, what can we do with that information? And then looking at different sites of the body. So can we look at vaginal microbiomes in a bit more detail? Can we look at oral microbiomes? Can we adapt the Labskin model to support these microbiomes that little bit more? We know that we can use synthetic body fluids, so we can use synthetic sebum, synthetic sweat to create different areas of the body. So we can look at things like armpits and groins versus dry areas like the back of your hands. So this is where we want to focus a little bit more. So in summary, the skin microbiome is a complex community that live on our skin and plays an important role in our overall health and well-being. Many of the factors that influence the skin microbiome need to be considered in skin disease. And understanding how the skin microbiome can be influenced may lead to new innovations in skincare and personal care. So I think we're going to see a lot more research and novel products in this area. Thank you so much for your time today. It's been an absolute pleasure to take you on a whistle-stop tour of the skin microbiome and Labskin. And if you have any questions, please do reach out. Thank you.